Good afternoon. Thank you everybody for being in my talk today. And today I will talk about drift selection, interactions drive, the diversification of Pitcairnia lanuginosa, bromeliad across its wide range, and structural neurotropical distribution. So to start, one second. It's fine now. So to start, I would like to thank our institutions that we could develop this study, the Brazilian, the French institution, and the Peruvian institution. The results I show you today is part of a PhD thesis of Barbara Leal. She has a poster tonight, and I would like to invite you all to come to the poster if you want to talk about some hybridization in orchids, and even about our results here. So today, actually, I want to open my talk with a very broad question of why there are so many species in neotropics. It's quite a broad question. And also, what evolutionary process have generated and helped to maintain the hyperdiverse biota, neotropical biota? To understand to answer those two very broad questions, we choose one plant family, the Bromeliaceae, which is a well-recognized, very good example of adaptive radiation in neotropics. We have almost 3,500 species in 75 genera, diversified super sampling less than 20 million years. I would say most of the hyperdiverse genera are diversified less than 10, 15 million years. And in thinking about the Bromeliaceae, what we want to understand is why bromeliads have diversified and widespread in neotropics in such a short time, and what evolutionary and ecological factors are involved in species persistence and coexistence in a highly diverse neotropical community. To answer those two also broad questions, we want to understand how population level process, gene flow, genetic drift, and natural selection affect diversification rates and emerging microevolutionary patterns in this neotropical rich family. And for doing that, today we brought a piece of the studies we have done in my lab, and we want to understand here the interaction between drift and natural selection in a naturally fragmented population of Pitcairnia lanuginosa. Pitcairnia lanuginosa is continentally distributed in two different biomes in South America, in the neotropics. So one is the Cerrado, the savanna biome, and the other is Yunga, Amazonian Andean transition biome, umbrophilous forest, very humid. And with that, we hypothesize that strong drift will happen due to reduced gene flow and low effective population sizes, and divergent selection will happen due to local adaptation due to divergent habitats. And so, to introduce who is this Pitcairnia lanuginosa species to you, our species is very uniform morphology, have a very uniform morphology across its wide range distribution. We collect individuals from the population and keep them in the greenhouses, and we could see them, they are very similar, and across the years that we have now more than 4 years acclimatation, they keep this similarity, this uniformity. This species is adapted, they prefer mesic habitats. They are citrus species, and they like to live in a riparian forest. But intermittent drought make them to have important adaptation to drought. So they have this broad age range to allow them to persist during these severe droughts that we have sometimes in the Cerrado biome. So this species is considered a desiccation tolerant species. When we have this severe drought, they will lose all the green leaves, and they remain the tuberose rhizome here with this spine waiting until the water comes, and then they will produce more green leaves. And this will allow them to persist and survive in seasonal habitats for a long period of dry. And with that, what we sample and analyze in this study I will present today was genetic data based on dd-red sac, SNPs from dd-red sac. The colored population are those we collect. The black ones are the information from the GBIF and the species link. And we also collect phenotypic data, the ecophysiological traits, 11 ecophysiological traits, in the green populations. You can see here the green population where we collect the ecophysiological traits. And with this sampling, we want to answer mainly two questions today. Can low effective population size resulting from population isolation and high levels of inbreeding preclude local adaptation in this wide range distributed species? And what is the relative importance of isolation by distance and isolation by environment on the patterns of species genetic structure? And jumping directly to the results, what we see here is that we have very strong, extremely low genetic diversity within populations. In the red here, you can see polymorphic sites. I'm not sure if you can actually do that. Okay. Here you can see the polymorphic sites, that they are not homogeneous distributed, so there's no pattern of why the population or other have lower or a little bit more genetic diversity. Expected and observed heterozygosity was really low. Most of individuals actually were homozygotes, which give us a very high inbreeding coefficient. And this also is was a pattern that we could observe because the species is a spontaneous self-pollinated. They are cleistogamous, the flowers mainly open with the stigma full of their own pollen. And we also observed during the field work, the population was very small, and their genetic data gave us this very low effective population size, very low. So in agreement with this very low genetic diversity, we found a very strong genetic structure. The overall population divergence, the FST, the global FST, was very high, 0.70. And we found here actually two main structures. One was between the two biomes, the Cerrado and the Yungas. They were very strong divergent. And the second was within the Cerrado biome, the eastern and the western populations, which are phylogeographic pattern commonly observed for other organisms in this biome. So with these two results, I believe I can show you that the drift should be expected to be very strong in these populations. And then we want now to analyze, to understand how are the signatures of natural selection. For that, we have three different approaches for studying, for understanding signatures of natural selection. The first were detection of outlier loci. We use BioScan and BioScan ANV using temperature and precipitation. And we found a very limited number of loci under putative divergence, only 2 here, only in the BioScan. In total, there was 34 outliers, and 22 of them were also detected with the BioScan ANV. Well, the second approach we used to identify signatures of natural selection was the genotype environmental association, and we use geographic pairwise distance matrices and environmental distance matrices to test the isolation by distance and isolation by environment against the matrices of genetic differentiation using neutral loci and outlier loci. We have those two things differently. And then we confront the models, including geography and environmental matrices, as a single predictor, and together a model using geography and environmental together. And for the neutral loci, what we can see is that the null model was the most significant, most important for the neutral loci, but for the outlier loci, what we found it was that the model that combined geography and environmental distance were the best, indicating that in fact isolation by distance and isolation by environment together could have a hole in the genomic differences of the outlier loci. And our third approach was a comparison of the phenotypic and genetic differentiation. And we use here the PST and the FST. The PST is an approximation of the KST when we don't have the additive genetic variation across anything population. And for the phenotypic differentiation among populations, we use ecophysiological traits. I won't talk too much about the ecophysiological traits now. If you want to talk, we can talk later. But basically speaking is that we, in the greenhouse, we submit some plants to drought tolerance, to drought stress, and then we collect 6 traits for the drought stress. And some plants will keep with a normal watering procedure, and then we impose here heat stress and light stress, and then we collect 2 traits of heat stress and 3 traits of light stress. And then we use those ecophysiological traits to calculate the PST using the R package and P-stat. And we confront that, we test that against the FST from neutral genetic differentiation among populations. And what we have here is a result in this panel showing the 11 ecophysiological traits, and you can see here the FST and here the PST. And what we can see actually is that most of these 11 traits were not significantly higher, neither lower than the PST, showing that neutrality is probably driving the evolution of those traits. But 2 of them, we could see that they are higher, the PST were higher than the FST, pointing to a divergent selection, and suggesting then the selection is related to drought tolerance in these populations. So, for concluding, what we see here, I hope I have shown you that we have a very low genetic diversity, very high genetic structure in breeding, low effective population sizes, all pointing for a very, very strong genetic drift. So drift is a really strong power in the evolution of these populations, but we also could find some divergent selection on few ecological traits, and this is in agreement with the combined effects of isolation by environment and isolation by distance we found. So together, actually, those results point for a joint whole of the drift that's very strong, but also natural selection in generating diversity and diversification of these naturally fragmented species. And with that, I would like to thank you very much and invite you again for the poster of Barbara tonight.
How does the seeds spread?
The seeds are in pairs. They don't have any appendage or anything. They are winged, but actually they are not winged. They are non-appendage dispersal. They basically fall.